Well, again, welcome everyone to the APS seminar series. Today, the moderator for today, we just received word she had to go out on an emergency issue, health issue, so I will make the introduction today. So we're delighted to introduce our speaker today, Liesl Bower-Jernigan. Liesl is a second-year PhD student, is a member of the Neil Anderson Lab, originally from Minnesota. She obtained a bachelor's degree in biology from Seattle University in Seattle, Washington. She didn't discover her passion for plants until the last 12 weeks of her undergraduate degree. She was researching populations of moss on the Seattle campus and then took a plant taxonomy class, and the combination of these two things, research in plants, led her to explore the option in plant sciences. After finishing her undergraduate degree, she worked at the Bailey Nursery in the inventory department, managing millions of plants. A year later, she ended up here at the University of Minnesota studying plant breeding and floriculture, which she'll talk to us about today. A little personal background. Outside of school, she loves to read, hike, and does some rock climbing as well. So we'll, as always, hold questions to the end, and you'll have the opportunity to come on and ask your question live, or you can raise your hand and to do that, and then you can also put a question into the chat as well. So with that, please go ahead and with your presentation today. Thank you.
All right. Thank you, Don, for the introduction. So the title of my presentation is Effects of Photoperiod and Saline on Flower Induction in the Chrysanthemum Arcticum Complex. So basic outline, I'll start with the background of chrysanthemums and a background of the Arctic daisy, and then I will go into my experiments. Chrysanthemums were the top selling herbaceous perennial in 2019 in the US, totaling $150.1 million. Breeders have been exploring and improving the ornamental traits, including plant architecture, floret types and size, early flowering time, medicinal compounds, winter hardiness, and many more. This photo is a photo of a winter hardy ground cover mum that was bred here at the U. And just some basic background about the breeding of chrysanthemums at the U. The University of Minnesota is the oldest chrysanthemum breeding program in the world, and it is the only institution breeding mums in North America. So some background on cultivated chrysanthemums. They are Chrysanthemum grandiflorum, and they are an interspecific hybrid, and it's believed to be derived from about 10 species. No one knows exactly how many because of the thousands of years of breeding that has been performed. They are allohexaploid with an average of 54 chromosomes, and I say an average because they are aneuploidy, so they do lose or gain a chromosome pretty common, and it can cause flower color changes. They are self-incompatible, so they produce very little seed, and they have low fertility, which leads to high breeding depression. So with that, they are primarily vegetatively propagated, and they are a short day plant flowering under 8 hours. Chrysanthemums were cultivated as early as 500 BC by Confucius, and the spread of cultivated chrysanthemums from China to neighboring countries began in about 400 AD. They were first introduced to the Western world via the Netherlands in 1688, but those plants disappeared. So in 1789, they were reintroduced into Europe through France. And in that same year, they were also introduced to the US. It was believed that all chrysanthemum species were native to Europe and Asia until 1741. In 1741, Dr. George Wilhelm Steller was on an expedition to map the easternmost coastline of Russia, and during his exploration, he did discover the Bering Strait. He was the first European botanist to step onto American soil in the Arctic and subarctic regions, And along his trip, he did collect many plant samples. On his way back to Russia, he stopped at Bird Island, which is a part of the Aleutian Islands, which is a part of Alaska, and collected samples of Chrysanthemum arcticum subspecies Arcticum. He discovered the only native chrysanthemum species to North America. So there is very little information known about the Arctic daisy. If you look it up in journals, there's less than 5 journal articles published at the moment about it. So most of the information we have comes from Steller's journals, herbariums, and then there are some Arctic flora books, as well as the research that has been done in Neil Anderson's lab and the research we are currently doing. So there is a main species, Chrysanthemum arcticum, a subspecies, Chrysanthemum arcticum subspecies Arcticum, and another subspecies, Chrysanthemum arcticum subspecies Polare. They are all commonly known as the Arctic daisy. And the centers of origin and diversity are Arctic and subarctic North America, specifically Alaska. And the species distribution is restricted to maritime environments. So on the images here in red, the red circle is indicating the main species Arcticum, and it is found specifically on mainland Alaska. The green lines are indicating where the subspecies Arcticum is natively found. It does overlap in distribution with the main species along the southern part of Alaska, and it spreads down to some, but not all of the Aleutian Islands, and it is found on the westernmost island, Attu Island. And then there are 2 remnant populations of the subspecies Arcticum found on the Kamchatka Peninsula in Russia, and 1 population in Eastern Asia and Hokkaido, Japan. And the subspecies Polare is indicated by the yellow lines on both maps, and it is restricted to the Arctic regions of Alaska, as well as in the subarctic and Arctic regions along the shores of the Hudson and James Bays in Yukon, Canada. So since Steller's discovery of the Arctic daisy in 1741, the species had not been studied until 2017. It had, however, been collected for herbarium samples throughout the years. Dr. Neil Anderson, my advisor, heard the Arctic daisy mentioned at a chrysanthemum conference in China. The Chinese researchers were interested in the species because they had not been able to collect it. They weren't granted permits to come collect the species in North America. So Neil decided he wanted to go collect it and study it without knowing anything about the species. So he went to herbaria and found the samples were collected from 25 locations. And then he went to those 25 locations to collect samples of it and didn't find them. So he did discover that the range of distribution decreased significantly from when it was first discovered. In the summer of 2018, Neil collected the main species Arcticum and the subspecies Arcticum from mainland Alaska and from Attu Island, respectively. So in this image here, you can see the red dots are where the main species was collected on the mainland, and then you can see in the orange dots where the subspecies Arcticum was collected on Attu Island. The main species was collected from 9 sites or populations, with varying numbers of genotypes collected from each site, with a total of 225 genotypes collected. And the subspecies was collected from 21 sites located along the southeast coastline of Attu Island. This was the only area accessible by boat to the shoreline and the only sites that are allowed by the U.S. Fish and Wildlife Service collection permit due to it being too dangerous because it's where the Bering Sea and the Pacific Ocean meet, so the weather is pretty bad. So there was a total of 326 genotypes collected. Then in the summer of 2019, the subspecies Polare was collected along the coastline of the Hudson Bay in the space indicated by the yellow circles. And 20 populations were collected with a total of 375 genotypes. The native habitat of the Arctic daisy is it does grow on cliff faces by the ocean or adjacent shoreline areas. So the image on the left is showing how close it is to the water. In that image, it's less than 25 meters from the shoreline. And the image on the right is just showing it growing on a cliff in between rocks. And it does also grow in brackish water. Brackish water is slightly salty water, usually where the mixture of river water and seawater meets. And the traits of interest that we are focusing on for the Arctic daisy are salt tolerance and cold tolerance. So an example for the salt tolerance we're interested in is in Minnesota, we use a lot of salt in the winter on our roads. And that salt ends up right on the sides of the roads in people's yards. So it could be a good use for that. And then the cold tolerance, there aren't a ton of few cold tolerant chrysanthemums. And for plant collection, the species were collected as rhizomes. And once they made it to Minnesota, they were transplanted and rooted in the greenhouse and grown under long days. And they did undergo some cooling treatments as well. And the survival rate for the main species and subspecies Arcticum differed greatly. So after the first 2 years in the greenhouse, the subspecies Arcticum had 0% death, while the main species Arcticum had a 36% death. And then for the polar species, after its first year in the greenhouse, it had a 22.7% death. So when I first started in the summer of 2019, there was a clear distinction between the main species and the subspecies Arcticum as seen by these images. So on the left is the subspecies Arcticum, and you can see that it has, this specific genotype is in bloom at the moment, but it was much bigger compared to the image on the right of the main species in the smaller pots, and they really weren't growing much. And the difference in growth was very apparent, as well as the flowering. So after the first 2 years, the subspecies Arcticum had 100% flowering in each year, where after the first year, the main species had 0% flowering in the first year and 3.1% flowering in the second year. And after the first year for the subspecies Polare, there's a 3.5% flowering. This image is actually of the main species. So you can see that they've grown a lot. They've filled out a lot more of their pots, and you can see that a lot more of them are flowering. So some previous research done in our lab by Yunjia, who was a master's student who worked on the Arctic daisy before me, was she looked at SNP markers to illustrate the genetic diversity and genetic structure of relatedness and variation among and within the species and populations. So this figure is showing us the relationship between the main species and the subspecies Arcticum. So the red is group 1, and it's indicating the subspecies Arcticum, and the green is group 2, indicating the main species. And so they did separate, with very few genotypes having SNPs that were shared. So it did show us they were significantly and distinctly separated as taxa. And it makes sense due to the distance between them of where we collected them. So it wouldn't make a ton of sense for them to share SNPs. And then next, Yunjia looked at within the main species populations, and she found that there was a separation of 2 groups. So in red was the population Old Valdez, and in green was the other populations. And again, there was very little overlap or shared SNPs between the 2 groups. And that makes sense because there is a geographic separation. This orange line is indicating a mountain range. The next analysis she looked at is the relatedness within the subspecies Arcticum. And it wasn't quite as a clear separation between the populations on Attu Island. So it did separate into 2 groups and then 2 subgroups within each, but there were a significant amount of shared SNPs. And again, that was due to the proximity of where they were collected. Yunjia didn't do any analysis on the polar species because it hadn't been collected, and then we hadn't gotten the data back for our SNPs. So while in Alaska, Neil did collect soils. So he collected from all of the mainland Alaska locations, and he collected one sample from Otto Island, and he only collected one due to a weight limit restriction on the boat he was on in the Bering Sea. So the soil samples revealed extremely high levels of sodium, as I am showing here in this table. They did also show high amounts of heavy metal in all of the populations. And the sodium concentrations in all of the populations collected were many levels of magnitude greater than the recommended greenhouse soil standard, which is 0 to 10 milligrams per kilogram. And we weren't able to collect a soil sample from the polar species due to restrictions of what can cross over the border. While Neil was in Alaska, he did collect flower heads, specifically from mainland Alaska, and brought them back to Minnesota. The subspecies Arcticum were not collected because they were not in flower during collection. And then the subspecies Ploidy was not collected either due to an import permit not granted. There have been a few publications that do include the ploidy of the Arctic daisy, but they do have conflicting results. So one of the papers said that it is diploid and the other said it is octoploid. So the focus of my research is going to be to characterize the main species Arcticum's flowering mechanisms, to characterize its winter hardiness, determine the main species salt tolerance and its effect on flowering, and to determine the ploidy levels of all the species and subspecies. So next I'll be going into my experiments. To start off, I'm going to talk about where most of my plants came from. As I mentioned before, the main species is pretty slow to grow. It's been doing much better, but cuttings don't root as fast, and it doesn't grow enough for us to take cuttings to have replicants for experiments. So for most of my experiments, I am using open pollinated seeds from the flower heads that were collected of the main species in Alaska. In the spring of 2019, 205 half-sib families were germinated and labeled by germ week. In our lab, we use colored toothpicks to indicate the germ week. And over 4,000 seedlings were grown, and then selections were made based off of each experiment, which I will discuss for each experiment. So one of our first questions was, what is the photoperiod of Chrysanthemum arcticum? To answer that, we did a photoperiod study. So our objective was to determine the photoperiod. And we did select 2 subsets from each of the 9 populations from the main species. And they were selected for early germination in weeks 1 and 2. So that resulted in having 6 to 36 half-sib genotypes per family. And we did choose a 16-hour long day and an 8-hour short day, and we separated them into those experiments and had them under those conditions for 185 days. And the data we collected was plant height, leaf number, number of shoots, internode length, inflorescence height, number of days to visible bud date, and flowering date. And we did find that it was a long day species. There was 0% flowering in short day, and 27 genotypes flowered in long day, with their parent plants being from Nilichuk, Anchor Point, Kenai 1, and Kenai 3. These images are examples of what it looked like in each experiment, each condition. So the long day genotypes, most of them were pretty large and were starting to grow out of their pots. This specific genotype had flowered. And 99% of the short day genotypes looked like the pot on the right. So after 185 days, there was a 13.2% death rate in the long day and a 92.8% death rate in the short day. And then after 345 days, there was a 99.9% death rate with 1 plant surviving in long day. And to this day, it is still alive under short day conditions. And that's a picture of what it looks like. This picture was taken about a week ago. So in summary, for the photoperiod experiments, Chrysanthemum arcticum is a long day plant, and we do plan to do future photoperiod experiments using 12 hours, 24 hours, and 12 hours, 20 hours, and 24-hour photoperiods, again using half subfamilies of the main species. So our next question was, what temperatures can Chrysanthemum arcticum survive in Minnesota? So to do that, we did focus on 2 experiments focusing on winter hardiness. So 68 open pollinated half sib Chrysanthemum arcticum families were chosen based on, we chose a sample of subpopulations from all of the 9 locations. And then we did choose half sib families that had seedlings germinated from 8 to 48 seedlings, just based on the numbers in the subpopulations. And then we did take 18 seedlings from 46 subpopulations and removed them for a freezer experiment. And then the remaining of those plants were used for field trials. So our field trials, we were focusing again on winter hardiness and flowering. So our objective was to determine winter hardiness and collect flowering data. So it is a 3-year field study, and our 3 locations are Rosemount, which is zone 4, Morris, which is zone 3-4, and then Grand Rapids, which is zone 3. So for those 3 locations, we planted at the research and outreach centers connected with University of Minnesota. So in Grand Rapids, we planted in week 41 of 2019, and then in Rosemount, we planted week 42 of 2019. And Morris, we planted week 21 of 2020, which I'll explain in just a few slides. And we had 291 or 292 plants at each location. And then we did have a completely randomized design. So this is an image of what the planting looked like in Rosemount on the left, right after it was planted in October of 2019. And then the image on the right is one of the flowering genotypes from this summer. And the data we were collecting is after overwintering, we're looking at if first what the plant looked like. So was it normal, have some injury, weak, meaning only 1 to 2 shoots, or if it died. And then once it flowered, we were collecting plant height, plant width, flower diameter, disc diameter, number of flowers, are the petals gapped or full, and other notes. And then we continued to mark if it, or for how long it flowered, checking once a week. And the same thing is done in Grand Rapids. The difference is that the North Central Research and Outreach Center staff are collecting that data for me. So the collection, or the picture on the left is a picture right after it was planted, and then a picture on the right is what it looked like this summer. So for Morris, we planted in May of 2020 due to poor field conditions in the fall of 2019. The field was too wet for it to be tilled. So the photo on the left is a photo of right after the field was planted. And then the photo on the right is about a month later. So you can see some of the plants are growing and it's looking pretty good. But we did run into a few issues. So due to COVID, there wasn't enough staff in Morris to do a weekly data collection for us. So instead, I went up once a month and just collected data on whether the plant flowered or not, looking for plants in bloom or things that I'd previously flowered. And we also discovered that this field plot is in a corner where there's a lot of water runoff from the rain, and they did get a lot of rain this summer. So you can see the picture on the right. It's really hard to distinguish what is actually the plant, and that's because they just got covered by mud. But we do have some preliminary data about flowering. So we did, or in the field, we have a much higher flowering percent than in the greenhouse, with a pooled percent of 41.2% and almost 50% in Grand Rapids. And with the data, we did run a 1-to-1 and 1-to-3 chi-squared test. And so far, 2 of the sites, Rosemount and Morris, show that location and flowering are dependent with a 1-to-1 ratio. And then for a 1-to-3 ratio, Morris and Grand Rapids show that location and flowering are dependent. And when it was all pooled, both 1-to-1 and 1-to-3 show location and flowering being dependent. So the second experiment we are doing for winter hardiness is a freezer experiment. So the objective is to determine survival at a range of soil temperatures. So again, we are using 46 half-sib families, and they either have 15 or 18 genotypes. And the temperatures for the soil we chose are 0 degrees, -4, -8, -10, -12, and -14. And so not all the half-sib families obviously have 18 genotypes. So we did go to -14 for the ones that had enough genotypes available. And we chose these soil temperatures to first have a range of temperatures, and then we specifically chose -10 and -12 because they are the lowest soil temperatures that zone 4 and zone 3 hit respectively. And then we chose -14 just to see how they would respond to an even lower soil temperature. So there are 4 runs due to space in the freezer. So you can fit 192 pots per run. And then within the runs, all of the genotypes were randomly chosen for each run, and then they were randomized within the run with 8 probe locations staying the same. And those probes are collecting the soil temperature. And then at each temperature, 3 plants per half-sib family were removed at each temperature, and those were also completely randomized. So this is just a graph of what the probes should be picking up in the pots. So what happens is we have a 2-hour ramp time. So the freezer gets 2 hours to reach the air temperature we want. And then there's a 10-hour soak. So the soil or the pots with the soil can sit at that temperature. And after that 12 hours, then 3 plants are removed. And on the left, I do have what the air temperature is for each soil temperature for my size pots. And again, 3 plants are removed from each half-sib family at each temperature. They are then placed back into the cooler until they thaw. And for me, it's taken about a week. And then after that, they are placed into a greenhouse to look at shoot number, shoot length, leaf number, whether they lived or died, and then rhizome data, which will be collected after 1 month of them growing in the greenhouse. And to explain why they're in these plastic bread bags, too much moisture was being released into the freezer, which was then ending up on the coil in the freezer. And so the freezer then wasn't reaching the correct temperatures. So we put all of our pots into plastic bags to prevent or to minimize the amount of moisture in the air. And then there are colored pieces of tape on almost all the bags, and that is for me to easily identify which pot needs to be removed at what temperature. So in summary, we are comparing 46 half-sib families that are both in the freezer and in the field studies. And then we are collecting flowering data in the fields, and we're hoping this will help us determine the winter survival of the main species. So our next question is, what is the ploidy of Chrysanthemum arcticum, its subspecies Arcticum, and its subspecies Polare? So for a ploidy measurement experiment, our objective is to determine the ploidy of all the species and the subspecies. So as I mentioned before, there are varying reports of what the ploidy is for the Arctic daisy. So it was mentioned that it was diploid or polyploid, specifically octoploid. And we have done some crossing and found it to be highly self-compatible. Some of our crosses or selfs did result in over 1,800 seeds. And then diploids are only the only self-compatible mums are diploids. So we are thinking this is possibly a diploid based off of that. So to help us determine the ploidy, we are going to use flow cytometry. Specifically, we'll be using a BD Accurai C6 plus flow cytometer system, which will be arriving this summer. And the basic procedure for flow cytometry is, first, you have to have a known standard. So a common one that is used and that I plan to use is a pea plant because it is diploid. And then what you do is you stain nuclei from cells with the fluorescent dye that binds to DNA, and then you run it through the flow cytometer. And what you get out is a histogram. So what you have to do is you have to compare the histogram. For me, it will be of the Chrysanthemum Arcticum to my standard to determine the ploidy. So I have a figure below, which was from a paper that was looking at 10 unknown ploidy chrysanthemum species. So in figure A is a known control. It is a diploid chrysanthemum. And figure B is an unknown ploidy. So when looking at the relative fluorescence intensity between A and B, you can see that they peak just about at 50. So them peaking at the same point does tell you that figure B is a diploid. So to summarize what I'll be doing for my ploidy experiment, it's to determine the ploidy of all the species and subspecies. And we think it may be a diploid based off of its ability or its self-compatibility. And this will help us with future breeding with other chrysanthemum species to know what it'll cross easily with. So our next focus question was, how does salt affect flowering of Chrysanthemum arcticum? And then what levels of salt can Chrysanthemum arcticum tolerate? So to test the salt tolerance of the main species and to see if its flowering is initiated by salt, we are using a hydroponic system. So I did do a trial run, and for this trial run, I did use a subspecies Arcticum, and that was because I had a lot of extra cuttings. So it was a good chance for me to use a hydroponic system for the first time. And we did measure our salt concentration using electrical conductivity. And we had 5 towers, which you can see here. 2 of them are kind of hidden in the back, but this is what they did look like. In the first tower, we didn't have any salt. It was a control, but our control did have fertilizer in it. And then we did increase the salt to 2.5 in the second tower, to 5 electrical conductivity in the third, then 7.5 and 10. And there really wasn't a reasoning behind the salt level. It was more for me to understand how the system worked. And the information we collected was leaf count, plant height, vegetative or flowering, leaf length, petiole length, and leaf width at the start of the experiment, 4 weeks into it, and then at the end, which was after 8 weeks. And we did analyze the data we collected and found that there was no significance due to the salt level, even though it was pretty high, which was kind of surprising to us. So for the next run, we will be using open pollinated half sib families that are from the main species. And then we will be using different concentrations of salt based on the soil data from Alaska, but we will be collecting the same data. So in summary of our the salt tolerance experiment is we're going to be using hydroponics to determine Chrysanthemum arcticum salt tolerance and its effect on flowering. So our last question for right now is, what is the genetic diversity and genetic structure within the Chrysanthemum arcticum complex? So our objective is to determine the genetic diversity and genetic structure of relatedness and variation among and within species and populations of the Chrysanthemum arcticum complex. So just some basic background. A SNP is a single nucleotide morphism, And it is a genetic fingerprint that can be used for species and subspecies. So we did recently get back some SNP data from DART-seq of our polar species. And we did a basic analysis with the data we already had for the main species and then the subspecies Arcticum. And included in this data is 20 CA seedlings. We're just seedlings that were planted in 2020. And based off of this figure, you can see that there is a huge separation between the main species and both of the subspecies, which isn't super common when you're looking at the relationship between species and subspecies. So I will be focusing more on looking at what is happening within the polar species. But that huge separation does make some sense based off of a huge geographic separation between all three of them, whether it was mountains, land, water. And in addition to the polar species, I also have collected dried leaf tissue from the open pollinated seedlings that are in the field trials and in the freezer trials. And they're in the process of being sent to DART in Australia, who will sequence it for us and then send us back the SNP information so then we can analyze it. So the genetic analysis summary is we will continue analyzing the subspecies Polari SNP data, specifically looking between the species and subspecies, and then looking at within the subspecies. And then we will analyze the open pollinated seedling SNPs and compare them to the parent plants. So in summary, Chrysanthemum arcticum has some major differences from cultivated chrysanthemums. It is a long day plant versus a short day plant for the cultivated mums. It is self-compatible. And it's possibly a diploid or an octoploid. And the interesting traits we are focused on are the salt tolerance, and then its winter hardiness. And again, the focus of my research is to characterize the Chrysanthemum arcticum flowering mechanisms, characterize the Chrysanthemum arcticum winter hardiness, determine the Chrysanthemum arcticum salt tolerance and its effect on flowering, and then determine the ploidy levels of all of the species and subspecies. And I would like to thank my funding sources, which is the Minnesota Agriculture Experiment Station and the Neil Anderson Flower Breeding and Genetics University of Minnesota Foundation. And I'd like to thank all of the Anderson Lab teammates that have helped me with my research. And then specifically at the North Central Research and Outreach Center in Grand Rapids, I'd like to thank Crystal, Keith, and Rosemanita. And then at the West Central Research and Outreach Center in Morris, I'd like to thank Steve Poppy. And at the Rosemount Research and Outreach Center, I'd like to thank Blake Webster. And then I'd like to thank Kelsey, who helped me with this great format for PowerPoint. And does anyone have questions?
Thank you very much, Liesl. So yeah, it's open for questions. You can raise your hand, and I think I noticed that Emily Hoover had raised her hand earlier. So Elizabeth, could you open it up for her to ask her question or make her comment?
I didn't actually ask a question. Sorry if my hand went up, but you did a nice job, Liesl. Thank you.
Thanks, Emily. I do see one in the Q&A. It says, any ideas of what mechanisms the species uses for tolerating or excluding salt and heavy metals? I don't. It's something we're looking into, but we don't have any idea at the moment.
Any additional questions? If you do in fact find that you have salt tolerance, you were talking about road rights of way, and here in the metropolitan area, everyone is doing these curbside plantings. Do you think you would have possibilities? Or how long? And again, how long does it bloom? Does it bloom for a day, 2 days, a week, a month?
Yeah, so we're interested in the salt tolerant gene is kind of what we're focused on, but it's to have that option for mums to be able to grow in like soils that have high salt. And it does flower, so it flowers in the middle of the summer, and there is a range of flowering depending on the genotype. I found that there were some that only flowered like a week and a half, or some had flowers flowering for a month. So it just kind of depends on what the genotype was.
So most of the mums that are in the marketplace, are they diploid?
No, they're allohexaploid.
Yeah, that's what I was thinking. Okay, will this necessarily have to be kind of a standalone genotype, or are there mums that are diploid that have value in the marketplace today?
There are, I know of one for sure that is a diploid. It's the Nankingensis. And I'm pretty sure it's on the market, but it would just add another aspect to it. So
Yeah, is it an annual?
I don't know off the top of my head.
Yeah.
Let's see. So Cindy asked, is it known what the native pollinators are? No, that's a really good question. We don't know. Actually, when Neil was in Canada looking for the polar species, I think him and I once were joking in a meeting about there were a lot of polar bears, and he had to obviously stay away from them because they're very dangerous. But we were joking that you never know, it could be the polar bears like rolling around that have pollen on them and pollinating it, but we don't know. So someone asked, what was the purpose of having 2 different winter tolerance tests? I imagine the freezer test was comprehensive enough since it covered a wide range of freezing temperatures. Yeah, that is true. We also are just curious how it grows in Minnesota because it's not native here. So having it in the field also opened that. But we also don't have that consistent ground cover. And so, or snow cover in the field. So yes, the freezer temperature helps with that, but then we can compare what happens in the field to what happened to in the freezer. Let's see. So someone asked, what is the breeding cycle for these species? Any compatibility with other genre? So we have crossed it with some other chrysanthemums as well. And there is some seed set. Obviously, with how little the main species flowers, it's we haven't had a ton of options to cross. But we did cross it with a Japanese chrysanthemum that was sent to us, and we did get some seed set for that.
Was that a diploid?
We don't know. It's like a trial one that they sent to us.
Yeah.
Let's see. So Vinny asked, how closely is this related to other chrysanthemum species? And how do you think it spread to North America? So that is a really good question. We are just studying this, so we don't know like exactly how related it is to other chrysanthemums. And we don't know how it spread to North America. I know that's not an answer people want to hear, but because it hasn't been studied before, we really don't know those answers. Let's see. Michael asked, I was wondering what salt you used for your salinity test, and if you're considering different salts for their effect on the plant. So for the, like just the trial we did, we just used regular like table salt. And I haven't considered using different salts, but that is a good thing to think about that I will think about for the next hydroponic experiment.
That's what I was just thinking. So what made the native soil salty? You know, have you guys done the analysis of the salt content in the native soils?
You're meaning like where they were collected from?
Yeah, the native sites.
Yeah, I had a slide with all of the exchangeable salt levels. And it's because they grow so close to the shore. They're within 25 meters of the water.
Yeah.
And then Neil did say that Chrysanthemum nancogenesis is a perennial rather than an annual. Let's see. So it was asked, are there chromosome bridges, crosses between species that could be used to allow you to introgress genes into the different horticultural lines? I don't know. For what I'm doing, that's not like we're not at that point yet. So I don't have an answer to that. Let's see. So John asked, in the light of the range of this species, is it possible that it once existed along the north coast of Alaska and Canada? Are you meaning like farther, like more Arctic? John, we need to unmute you, John.
What I mean is this species is a coastal specialist, right?
Mm-hmm.
But it's found along the Pacific seaboard and then over in Hudson Bay.
Mm-hmm.
So how did it get from how did it spread from one place to the other? The only sort of coastal route would be along the northern coast of Alaska and Canada.
Yeah.
Which would require, I think, a warmer time period than we had lately.
Yeah, that's a good question. We don't know how it traveled. And that is a reminder that's obviously the polar subspecies, but the guy who discovered it, Stellar, did do an expedition up along Canada, Alaska and Canada. So there were people that did travel. I know it's not great weather, but they did travel. Let's see. So Grant asked, how are seeds dispersed? I know, again, we don't really have a great answer.
Maybe the bear story.
Right. I mean, like, who knows? It could, go on to a bear. We don't know. Yeah. And then can you artificially cross the different Arcticums? Yeah. We've been crossing them when we have the ability to. Yeah.
Any more questions? Comments?
I think there was one question from earlier from Dom.
Oh.
That answered. That here you indicated there could be some application for these species on roadsides in Minnesota. What are these species? Relative mowing tolerance, height, frequency, etc. Roadsides also have other stresses, heat disturbance, etc. Do you have thoughts there? for these species to be competitive in a mixture?
Yeah, so they are more of a ground cover. And the roadside, it was just more of an example of what salt tolerance can be used for, specifically Minnesota. We aren't there yet. We're still trying to get this main species to flower consistently. So those are more of our focuses than at this exact moment what we're going to implement it for. So it really just depends on what we find out. And then we can decide how we're going to implement it. One question was, what discovery did you find most interesting and why? I guess for me, it's more of what I find the most interesting is Neil's stories about how he discovered it. So he went out to Alaska not knowing he would actually find the plant. He had some ideas of where it might be. And then he did find it, but it took him, he went on a whole adventure that, he was stuck on a boat for a week and it was really supposed to only be 2 days. And I think that's the most interesting part for me, is that it took a long time for him to find. Yeah.
Are there any other comments or questions from anyone? If not, thank you very much. A very nice presentation and a good exchange with the folks that were participating in the seminar today. So also want to thank all of those folks for the comments and questions that they brought up. Try to keep an active conversation as we go through these presentations. So I appreciate your participation as well. Thank you.